All right, here we go. This is session 10 of our 2023 Defending Genesis conference. And I'm just going to get right into it. I am going to share my screen and get our presentation going, as I have quite a bit to cover in the next hour. And so, again, this is session 10 of the Standing For Truth Ministries Defending Genesis conference. And okay, I just want to make sure everything's good to go. And it's been a great week. We've had a ton of great speakers, presentations, excuse me, and it's been highly informative. And that's what we like to do every year, is give our audience, give our skeptics and non-skeptic friends a lot of material and content. And we strongly believe in engaging the opposition, engaging challenges head on. And so that's what I'm going to be doing with my specific presentation, which is titled Dismantling Junk DNA or Junk Science. I'll tell you right off the bat, it's junk science. And I'm going to demonstrate that throughout this important presentation. And so let's move on here. Defending Genesis 2023, hosted by Standing For Truth Ministries. This specific presentation was put together by myself, Donny Budinsky, and I'm the founder of Standing For Truth Ministries. I am going to be focusing on a lot of the discriminatory lines of evidence in terms of the creation versus evolution debate. Firstly, I want to set the foundation for many of the conclusions I'm going to be making by going into some of the agnostic lines of evidence when it comes to the origins debate. And so stick with me, and you'll see by the end of the presentation as I build up my argument and show you why the junk DNA paradigm and everything within it, including endogenous retroviruses, pseudogenes, allele sequences, so on and so forth, basically what the evolutionary community claims are the best lines of evidence for evolution. I've written numerous books. One book here, Special Creation, and also the Endogenous Retrovirus Handbook. So one thing I've really enjoyed doing, and I think it's important over the last 4 or 5 years since doing this, especially over the last few years doing this full time, is I like to engage comprehensively what the evolutionary community argues or sees as the best evidence for common descent. And typically when you search the best evidence for common descent or evolutionism, you'll find endogenous retroviruses, chromosome 2 fusion, you'll find junk DNA, pseudogenes, genetic level nested hierarchies. And so these have been the main areas of focus for myself. We do have a team, of course, and we all have our various specialties and areas of focus. And I like to dismantle their very best lines of evidence. And what I've discovered in my active research projects is what they typically argue or put forth as the best evidence for evolution, that kind of evolution that says whales, pine trees, humans, and strawberries are related through common ancestry, is that these lines of evidence actually turn out to be some of the best evidence for creation, for the separate ancestry model. And I find that to be fascinating. And so with the overturning of junk DNA actually comes the overturning of several of the evolutionary community's favorite lines of evidence, like ERVs, pseudogenes, allele sequences. And so you can find a detailed refutation of endogenous retroviruses in my book here. I've debated it numerous times. I've given numerous presentations on the topic as well. Special Creation, I highly recommend this book. I get into all of your best data points and arguments for separate ancestry, in a literal Adam and Eve. I also touch on the chromosome 2 fusion, Neanderthals, human evolution, out of Africa versus out of Middle East, evolution versus devolution. I touch heavily on genetic entropy, the evolutionist favorite argument. And so I've also written a book on eschatology, End Times Revealed, Dawn of the Antichrist. You can find endless content over on our website, standingfortruthministries.com. You can find articles, books, videos, endless videos, and resources. Right on the main page, you'll find a whole large section answering objections and answering common questions. And you'll just have all of your most common questions and challenges. And just with a click away, you'll find an answer or a response. So I'd like to start off the presentation with some good quotes here. For example, I think we can all live by this for sure: believe God more and believe man less. The Bible says that we should not put our faith in the wisdom of man, but in the power of God. Man is fallible. God's word is our final authority. Holy men spake as they were moved by the Holy Ghost, the very words of God. And so believe God more and believe man less. Another quote: good science always affirms Scripture. And that is so true. When we actually look at the empirical scientific data, when we look to good science, not pseudoscience, okay, like for evolution, there's an equivocation fallacy. When the evolutionists say evolution, oh, you don't believe in evolution? What they typically mean is the theory of evolution or the theory of common descent that says all life, including banana plants, humans, whales, dogs, and cats, are related through common ancestry and basically go back to a single common ancestor billions of years ago. Okay, we're going to have an issue with that. That's not science. But if by evolution you mean change over time, if by evolution you mean changes in allele frequencies, an allele being a genetic variant, frequency being the frequency that variant or gene occurs in a population, that frequency can change over time, of course, then we're not going to have an issue. Change over time does not equate to banana plants and humans are related through common ancestry. My phone changes over time. Cars change over time. The way we think changes over time. The things we believe change over time. So we're not going to dispute that. Good science always affirms Scripture. The Bible claims to be the history book of the universe. What does the Bible say in Genesis? God created two people, Adam and Eve. Adam, the first man, Eve, the mother of some living, all living. We can test these claims through scientific data. And what do we find? We find confirmation of the Bible's account of human origins. We've literally discovered our first parents in our DNA. We have evidence for one woman that we've all descended from. Mitochondrial DNA, that's the powerhouse of the cell. It's outside the nucleus, and it's a special piece of DNA, only about 16,000 letters long. It's small, and it's passed down on the mother's side. And so you can do a pedigree study where you get grandmothers, mothers, daughters, count out the DNA differences between them. And what you get is a fast mutation rate. So the mitochondrial DNA mutates fast, but there's also very low diversity. On average, there's only about 25 DNA differences that separate any two people on the planet. That's trivial. That's biblical time. How long does it take to accumulate these few DNA differences with a fast mutation rate? Not much time at all. There's not a couple 100,000 years worth of DNA diversity in the mitochondrial genome. Mitochondrial DNA worldwide can be traced back to a single female ancestor in the not so distant past. Y chromosome. That is nuclear DNA, but it is uniparentally inherited DNA, just like the mitochondrial DNA. It's not biparentally inherited DNA like autosomal DNA. So these DNA compartments lack recombination. Y chromosome, there's some evidence, a little bit of recombination at the ends, but so they're less messy. They're passed down largely unchanged and more stable. And so your Y chromosome is passed down on the father's side. So you can do the same. Grandfather, father, son, count up the differences. You get a very fast observable mutation rate. in the Y chromosome, it's about 1 to 3 per generation. In the mitochondrial DNA, it's about somewhere between 0.1 to 0.5 per generation. This is fast, far faster than the evolutionary community ever predicted. And so you could take Y chromosomes worldwide, male Y chromosomes worldwide. Okay, I got my Y chromosome from my dad. My dad got his from his dad. And so it's this unbroken line. You can trace back all male Y chromosomes on the planet to a single Y chromosomal ancestor just about 4,500 years ago, to Y chromosome Noah. And so we've discovered, this is just one example of many. I've given entire presentations on this, where we have evidence for two people that we've all originated from or descended from. Take your mitochondrial DNA. So you have Eve has kids, her kids have kids, and as mutations accumulate, you get new branches on the family tree. Take those branches back, okay, start taking them away back to an original point, and that would be Eve. That's what we find. The mitochondrial DNA is nothing like chimpanzees. The Y chromosome, nothing like chimpanzees and humans, that is. And so there's separate ancestry. There's one example of where the Bible says God created two people, Adam and Eve, the mother of all living, the first man. And science has confirmed this. And so this is why it's important that we understand that good science always affirms good scriptures. So I've done presentations addressing all of the best so-called lines of evidence for evolution: endogenous retroviruses, chromosome 2 fusion, pseudogenes. Today, we're going to be dealing with junk DNA. Now, to set the foundation, though, for our talk here on junk DNA, I want to go over the separate ancestry model. The separate ancestry model versus the universal common ancestry model. So the universal common ancestry model, as we know, says all life is related through common ancestry, including apples and whales, plants and animals. It was Darwin who said it is a fact that all life, including plants and animals, should be related. The separate ancestry model says rather than everything being related, there exists separate ancestries, independent origins, where there is ancestry, yes. Some animals do belong in the same lineage or in the same ancestry, but other organisms are separate. For example, here we have humans that our model would suggest are unrelated to any other form of life, including the chimpanzees. So humans were created independent and separate from any other form of life, specially created. That's the separate ancestry model. So humans, if they are not related to orangutans, chimps, gorillas, right? That's the great ape family, kingdom, phylum, class, order, family. So the great ape family, genus, species. The evolutionary community, evolutionists, taxonomists, have humans within the great ape family. We would have humans outside of the great ape family. Homo sapiens belonging in their own group altogether. And we've actually demonstrated this. Again, I've done many presentations going over the Y chromosome, the mitochondrial DNA. For example, the Y chromosome between humans and chimpanzees, they're nothing alike. They're incredibly dissimilar rather than similar. When you consider overall gene content, architecture, and size differences, the human male Y chromosome is only about 35 to 40% the same as the chimpanzee Y chromosome. As a matter of fact, when you bring in the gorilla Y chromosome, remember the evolutionists say the chimpanzees are supposed closest cousin. When you bring in the gorilla Y chromosome, humans and gorillas have a more similar Y chromosome than either are to the chimpanzee Y chromosome. Well, there's a break in their so-called nested hierarchy. And there's no way to account for such massive size differences. You'd have to explain the evolution of entire new gene families, chromosomal reorganization, and massive loss of gene and gene content if humans and chimpanzees are related. And that's just one of the many lines of evidence that demonstrate humans and chimpanzees aren't related. There's an interesting class of genes called orphan genes. They're these taxonomically and essential genes. And you find the certain orphan genes in humans, certain orphan genes in chimpanzees, gorillas. They're not shared. So by orphan, what that means is you have them in humans and chimpanzees, but we have no evidence for their precursor genes. What did they evolve from? They're just there. They're specially created. They pop up out of nowhere. Evolutionary rescue devices don't work. And this is what we would expected under the separate ancestry model, that there would be unique genes, unique DNA elements spread out across the different kinds of life. And their arguments involving what's called de novo gene synthesis, very circular. They'll invoke something called de novo gene synthesis to explain these orphan genes. Why it's circular is because the evolutionists, rather than admitting, okay, this does look like separate ancestry, they'll say, no, orphan genes exist because of de novo gene synthesis. These genes must have popped up from non-coding areas of the genome through mutations and other processes. And then they'll say, well, why de novo gene synthesis? Why invoke that? Well, because we have orphan genes. Do you see this circle? Orphan genes because of de novo gene synthesis. Why is de novo gene synthesis a thing? Because of orphan genes. So they just argue in a circle rather than tapping out and admitting, yeah, this does look like separate ancestry. So here's the universal common ancestry model, where all life, including crocodiles, birds, snakes, mollusks, mushrooms, ferns, bacteria, all go back to a single common ancestor. You got humans at the top, chimpanzees, and you'll notice how this is a branching tree. And so the branches all do connect. And so the evolutionary model has humans and millipedes related, humans and insects, humans and mushrooms. You're related to a mushroom. That spaghetti meal that you ate with mushrooms, that's your cousin. You're related through common ancestry, according to the evolutionary model, where we would argue that, yes, there is relationship amongst various organisms. We would define kind, at least I would, according to my model, as groups of organisms that can be traced back to a single common ancestor. And that single common ancestor and the group itself is unrelated to other groups of organisms that go back to their own common ancestor. And I've demonstrated that. I've answered the phylogeny challenge using humans. Tree of life, again, all life, including whales, elephants, and butterflies, and dinosaurs, humans and birds, and leaves and octopus, all going back to a single common ancestor. Here's a more sophisticated tree of life. We are related to every living thing. What an assertion. What an assertion. Refuted by so many data points, especially in the world of genetics. Remember, what's the best way to answer this question of ancestry? It's through genetics. What's inherited? Sperm and egg. Genes, traits, genetics. So this is the most direct way to answer the question of ancestry. What's not inherited? Sperm and egg? A rock, geography, geology, genes and traits. And that's why it is through genetics that common descent has been decimated. I have had 100 formal debates and probably a couple hundred impromptu open mic debates and discussions. And so I've had plenty of engagement with the opposition, and they lack any real credible or sophisticated response to these major data points like the Y chromosome, the mitochondrial DNA. No amount of rescue devices can save the day for common descent. So if by evolution you mean coyotes, domestic dogs, wolves, and dingoes are related through common ancestry, if by evolution you mean changes in allele frequencies in populations over generations, sure. We're not going to have anything to argue over. Like I said, our phones change over time. Cars change over time. Technology change over time. Organisms change over time. Organisms adapt. See, here's the thing. Ever-changing environments require ever-changing genomes, or else we'd have nothing but extinction. Organisms have to be able to adapt. Even cars today are built with adaptive mechanisms, front-loaded, because man is slowly figuring out over time, being able to recognize the characteristics of design. But God has thought about this since the foundation of the world, and he's given animals and creatures the ability to adapt to various environments. Vehicles today have backup cameras. They've got alarms where if you're backing up too close to another car or curb, it goes off. lane control, where if you're drifting off into another lane, the car has a built-in mechanism to put you back in the same lane. If you're slipping in the winter, anti-slip mechanisms. And so all sorts of safety mechanisms built in, front-loaded. And that's what we find in living organisms, as well as a lot of front-loaded diversity, the front-loaded ability to adapt. There's a lot of morphological adaptability built into organisms, and that has to be the case. Now, if by evolution you mean domestic dogs, ponies, whales, and banana plants are related through common ancestry, throw mosquitoes in there, that's where we're going to have an issue. That's not science. That's fish to fisherman evolution. That's bacteria-like organisms to biologist evolution. That's a fairy tale. They think time is the hero of the plot for them. Time's not the hero. Time is the enemy. More time equals what? More extinction, more degeneration, more problems, less large-scale evolution, as a matter of fact. So let's go over some more or less agnostic lines of evidence for the creation versus evolution debate. Now, what I mean by agnostic is these are lines of evidence that can't really fully discriminate between the data points, between the models. They don't offer enough preclusionary data points. If we were debating creation versus evolution, and I said the sky is blue, therefore creation is true, or the evolutionist said the sky is blue, therefore evolution is true, you say you're nuts. Creation and evolution can both explain why the sky is blue. Many different models could. If I were to say, well, the earth is round because creation's true. No, both models, the evolution model and the creation model and many other models, can explain why the earth is round. We need to look at data points because there's a lot of overlap between the creation and evolution positions and models. We need to look to where there's not overlap, to where we have a point that only one model can explain and the other model cannot explain. I've already talked about a couple, orphan genes and Y chromosome. The evolutionists love to waste a lot of time on agnostic lines of evidence where, sure, this is the observation, this is the data point, this is the line of evidence, but both models can explain it. So we're going to briefly go through these as we set the foundation for dismantling junk DNA. Classification, systematics, homology, nested hierarchies, and so-called transitional fossils. Let's start with classification. So I'm sure everybody's familiar with Linnaeus classification, kingdom, phylum, order, family, genus, species. Okay, kingdom, phylum, class, order, family, genus, species. And you could group organisms. Sure. We can group organisms according to various characteristics. We can group all kinds of automobiles. If you go to a car lot, you're going to find SUVs over here, vans over here, crossovers over here, sedans over here, hatchbacks over here, maybe diesels over here, okay, non-diesels over here, gas engine powered vehicles. And it doesn't prove relationship is the point. My bookcase upstairs is largely organized by theology books. And then within theology books, soteriology, eschatology, you've got science-related books, sales-related books. So we all love tools. You can go to your work shed and you've got all your different tools grouped together, okay? It doesn't prove any kind of relationship. And we can do that with animals as well. And you're going to find that humans do share a lot more in terms of characteristics and morphology, anatomy, and physiology with something like a chimpanzee, in the same way that a sedan is going to have a lot more in common when we start grouping things based on various traits with a hatchback than a sedan and a hatchback would with what? With a boat or an airplane. And you could draw a circle big enough where you can group anything together. Things that breathe. Okay, now you got humans, gorillas, lions, bats, platypus. How about things affected by gravity? Okay, now you got all forms of life, but now you can put in a chair, shoes, a bowling ball. Doesn't improve relationship. It just proves we can group things based on various categories. Motorized transportation, sure. Boats, trucks, airplanes, motorcycles. And you can do some research and you can find classification of automobiles, and you're going to find based on the purpose of the vehicle, based on the kind of fuel used, and you'll find the exact same thing when it comes to the animal kingdom. That's why you have these various groups: fish, amphibians, reptiles, mammals, giving milk to live young, having hair, things that make someone a mammal. You got vertebrates with a backbone, invertebrates. Okay, there's various characteristics that would group the various creatures together. And it's interesting to do so, but doesn't prove relationship. Now, so-called homology, let's say. So you've got human, cat, whale, bat down here, and you're going to have similar structures in the forearm, similar structures or similarities in, the hand, similar bones, comparative anatomy. But at the same time, as the evolutionist says, well, these homologous structures, these similarities, homologous patterns, they're because of common descent. We've inherited them over time. But the creationists would say, no, this is due to common design. Look at automobiles. Okay. Sedans made by GM, made by Ford, Chryslers, Honda, Toyota, Volkswagen. They've got 4 tires. They've got steering wheels. They're built from a certain height from the ground. Various similarities in safety mechanisms, backup cameras, navigation systems. Generally, they have a windshield in the front. They've got mirrors, seats. Some share leather seats. And so some even share in terms of micro similarities. If you want to look at the materials used, let's say within the car, similar paints. So similarities does not prove common descent, and it doesn't necessarily prove common design. And in science, you're not really it's not about proving something. It's about disproving a model. What's the best explanation for something? Come up with a hypothesis and then make testable predictions. Here's my hypothesis. In light of that hypothesis, I predict this should be true. This should be the case. And try really hard to disprove your model, your hypothesis, and see if it withstands scrutiny and withstands time. Okay. And so human engineers build in homologous patterns. And so why shouldn't God? But even in homology, that is overall agnostic to the debate, because both models can explain it. There's actually many data points that make it not agnostic. So, for example, I like to say it's the differences that make all the difference. So, yes, even within the similarities in genes, let's say, between humans and chimpanzees, you actually have some major differences in the way those genes are expressed, the way those genes are turned on, the way those genes are regulated. There's differences in alternative splicing, differences in epigenetics, differences in the 3D architecture of these genomic systems. And so even in the similar regions of the genome, there's differences. And it's the differences that make all the difference. Sure, there's similarities in the Y chromosome between humans and chimpanzees, but guess what? It's half the size, the Y chromosome, between humans and chimpanzees. Major differences in architecture, gene content, size differences. And so, yeah, there's similarity, but there's too much dissimilarity for the evolutionary model to explain. And so, although the overall argument of homology is agnostic, when you actually start to dig deep and look at the finer details, it is a way to discriminate between the models for sure. You could also look at so-called transitional forms, like Tiktaalik, right? The famous fishapod, or Archaeopteryx. But the problem for the evolutionists is, can the evolutionary model only explain this data? Well, I want to move on here before I answer the transitional forms argument. Here we have humans building in homologous patterns, all kinds of similarities. We have something called hard to classify vehicles. The creation model would say, okay, we start from Genesis. We start from the Bible. The Bible claims to be the history book of the universe. The Bible says that we, as humans, we're made in the image of God. And therefore, there should be something about us that reflects the divine. And when we look to the biological world, we might be able to get a sense for how God designed based on the way we design, because we are made in the image of God. And it just so turns out that we build in homologous patterns, we build in nested hierarchical patterns, and we also design some hard to classify vehicles. With the invention of the crossover vehicle, they resist classification. The Jeep Gladiator, it's a blend of a truck and a Jeep. The Ford Flex, blend of a van and an SUV. They're called, it's not a van, it's not an SUV. If you're just looking for something that an SUV would do in a specific environment, it's not going to be perfect for you, or a van. But if you want something right in between, boom, you go with a crossover. The Dodge Journey, same thing. They resist classification. The military, they've designed something called a military amphibious assault vehicle. So if you want firepower just for the sea, this wouldn't work. You'd want just a military boat or ship. If you want something just for the land, like a tank, then this wouldn't necessarily be the best option or design. But if you want something for that transitional environment between sea and land, then the military amphibious assault vehicle is perfect. It's hard to classify. It's not necessarily a land vehicle. It's not necessarily a vehicle designed for the sea. No, it's designed for that transitional environment between sea and land. It's a hard to classify vehicle, but it's designed for what it's supposed to do. And so you can take the ship that's built for the sea, you can take the tank that's designed for the land, and you can stick that military amphibious assault vehicle right in the middle. And I can say, you know what? The military ship evolved into the transitional form that's known as the military amphibious assault vehicle, and then that evolved into the tank. No, but that's not true. Each vehicle is actually designed for a specific purpose, for its appropriate environment. In the same way that I could take a unicycle, a bicycle, a tricycle, and a four-wheeler and line them up and say that the unicycle evolved over time into the four-wheeler, that would not be the case. And that's what the evolutionary community does with a lot of these fossils. Fossils are fickle, and they'll tell you any story that you want them to tell. Okay? And so I could predict, I got a unicycle, a bicycle, and a four-wheeler. Oh, there must be a tricycle out there somewhere, a vehicle with three wheels, or something to get me from A to B, a tricycle. And yet we find a tricycle, oh, proof for evolution? No. Evidence for evolution? No. Even when it comes to Tiktaalik, they actually discovered tetrapod foot tracks, supposedly 20 million years, the evolutionary timeline, before Tiktaalik was where in the geological rock strata Tiktaalik is found. That means we had tetrapods existing before Tiktaalik. How is this a transitional form? It's an interesting mosaic. It's a hard to classify creature. We should not fear hard to classify creatures because, again, this fits the design hypothesis. If we're made in the image of God, the Bible tells us that we are, then we should be able to get a sense for how God designed based on the way we design. And we design in homologous patterns. And therefore, why shouldn't God? God thought of if we're just catching up to God. That's why just now we're seeing major car companies front load their vehicles with safety mechanisms. They're thinking ahead. That's foreknowledge. And that's what God has done when he's front loaded the ability of his creations to adapt to various environments. So we have hard to classify vehicles. We build in homologous patterns, nested hierarchical patterns. Here's nested hierarchies, these groups within groups patterns. The fact that humans share more with the chimpanzee than humans do with a dog or a fish. Humans can be grouped together with primates because we share more in terms of morphology, anatomy, and physiology with old world monkeys, new world monkeys, the great apes, so on and so forth, than we would with a whale. Okay, so we can be grouped in with primates, but then all the primates can be grouped into a greater group, mammals. A whale is technically classified as a mammal rather than a fish. We can all be grouped together as vertebrates. And so you draw a circle big enough and you're going to have everything grouped together. And so, sure, there exist these groups within groups patterns, the nested hierarchy of life. A human shares more with a chimpanzee than a human shares with a bacteria. But what does that say? Well, again, here, even within the so-called great apes or old world monkeys, new world monkeys, humans and the great apes share more with each other than they do with a lemur. Humans share more, as you can see here, with a chimpanzee than they do with a mouse or a lizard or a fish. But just stand back and look at the creatures. Right off the bat, you should be able to tell, yeah, a human does look a little more like a chimpanzee than a human does with a fish or a salamander. But I pointed out there's many inconsistencies in the argument from nested hierarchy, like the Y chromosome. The chimpanzee and the human Y chromosomes are incredibly different. As a matter of fact, the human and gorilla Y chromosomes are more similar to each other when you consider everything than either are to the gorilla or to the chimpanzee. That's a break in the so-called nested hierarchy. Anytime convergent evolution is basically, according to the evolutionists, the independent evolution or acquiring of these traits that are not actually due to homology, homoplasy instead. And so they'll argue these same traits evolved independently multiple times. Yeah, anytime that's invoked to explain an inconsistency, it's actually an admittance into the lack of uniqueness of the evolutionary phylogenetic tree. There's another thing called incomplete lineage sorting. Depending on what gene you look at, depending on what genetic marker you look at, you're going to get totally different trees. And so although there's an overall hierarchy, yes, human does look a lot more similar to a chimpanzee than it does to a mouse. When you start looking at the details, you start looking at the differences, because the differences make all the difference, then you see where all the inconsistencies are. This is where you see many of the discriminatory lines of evidence that exist. So notice we go back to the design hypothesis. Can we explain a general nested hierarchical pattern in the biological world? The answer is yes. Humans accidentally build in hierarchical patterns. Do you think major car companies like Chrysler, Ford, GM, Honda, Toyota, Nissan, Volkswagen, do you think they're going to the drawing board and saying, you know what, we want to design our cars in a way that reflects the biological world and the nested hierarchies that we find in the biological world? No, of course not. They accidentally build in hierarchies. When they built a sedan and an SUV and a tractor trailer, a semi-tractor trailer, by definition, you're going to have the sedan more similar to the SUV than either are to the semi-tractor trailer. Once you bring in an airplane or an unpowered vehicle like a bicycle, then you have a nice nested hierarchy where a sedan, all sedans share more with each other than if you bring in a hatchback. But then a sedan and a hatchback share more with each other than they would with a truck or an airplane. So you have this natural nested hierarchy within design things, just like we see in the biological world. And so human engineers build in nested hierarchical patterns. Human engineers build in homologous patterns. Human engineers design hard to classify vehicles. And this is also what we see in the biological world. It's amazing, incredible confirmation of the design hypothesis. Humans, being unique, would be their own group right here. But because we are still designed and built with DNA, RNA, protein, similar structures in terms of anatomy, the DNA coding for the anatomy, phenotype being a reflection of genotype. Genotype is the genetic content, phenotype being your outward features, physical characteristics. Okay. And so they're linked. And therefore, yeah, once you start looking at traits and classifying creatures, then of course you're going to have to put humans somewhere on the tree of life. And rather than grouping them in with bacteria or mosquitoes or dragonflies, they would technically be grouped together with the great apes. Because every creature that you look at has to, by definition, be more similar to one creature than another. Pick your creature, an elephant. Okay, your elephant's going to have to be more, an elephant is more similar to a rhino than it is to a lion or an octopus. Pick a brachiosaurus, one of your long-necked dinosaurs. That's going to be more similar to another type of dinosaur, like a T-Rex, than it is going to be to a fish, like a shark. So no matter what you look at, every creature has to be more similar to another creature than it is to other creatures. There's always going to be dissimilarities and similarities. Notice tools, different wrenches. You have hard to classify tools. Look at a fencing plier. Where does this fit? It's designed. It's not so black and white, is it? It's designed with, it has a multi-purpose design, staple puller, staple pincher, fencing wire cutter, all types of things built in, multifunctional tool. And so if you were to start classifying this, well, where are you going to put it? It's difficult. It's a hard to classify tool, just like we have hard to classify vehicles. And so the point is, these arguments from so-called transitional forms, homology, nested hierarchical patterns, they're overall agnostic to the debate. Now, because the differences make all the differences, once you actually dig deep and start studying the differences, you realize that within the homology argument, within the nested hierarchy argument, you find preclusionary data points that preclude the evolutionary model from being true. Because only the design hypothesis can explain the data. And so there are two major differences between the creation and evolution models when it comes to the origin of genetic variation and diversity. The evolutionary model explains the origin of all genetic diversity or variation as being the result of mutations over time. A mutation adds something new because it's adding something that was not previously there. And so over time, mutations accumulate and they allow for the necessary genetic variation for natural selection. It's a fine-tuning mechanism, keeps a species as strong as it can be. So natural selection can act upon this variation. That's how the evolutionary model goes. And you have gene flow, recombination, gene conversion, genetic drift, various mechanisms that the evolutionist looks to. But the creationists, we explain the vast majority of DNA differences and genetic variation as being the result of what's called created heterozygosity. Heterozygosity just meaning a state of DNA diversity. Homozygosity, a state of no DNA diversity. Goes back to basic genetics, basic alleles. Capital A, lowercase a, capital B, lowercase b. If you're capital A, lowercase a, you're genetically heterozygous at that genetic loci. Genetic loci being a genetic location. If you're capital A, capital B, you're genetically homozygous because there's no diversity there. It's just capital A, it's the same rather than being different, capital A, lowercase a. And so if you apply this to the millions of positions within the genome, capital A, lowercase a, capital B, lowercase b, you have the incredible potential for genetic variability in a single generation, just from recombination, where these chromosomes line up, exchange genetic material and information to create new chromosomal combinations every generation. If the DNA difference is already built in from the start, then you can get new varieties, new chromosomal combinations quickly. The DNA differences are built in. You don't need millions of years to build up the necessary genetic variation through mutations for adaptation to take place, for new varieties to come about. That's what the evolutionist model needs. Why this is important is this model called design diversity, where God designs creatures with internal diversity that can be utilized through processes like natural selection, gene flow, recombination, gene conversion to lead to new varieties. Morphological adaptability built into creatures that can become manifested as time goes on. And novel phenotypes can come about simply based on the pre-existing ability for that change, for that adaptation. Evolutionists treat two very different things as if they're the same. The evolution of higher life forms, large-scale evolution, macroevolutionary changes, and adaptation to environments as if they're the same thing. No, they're different. One happens, the other doesn't. Adaptation happens. Macroevolutionary change, on the other hand, doesn't happen. And even if you look at the change when we get speciation events, because the evolutionists will say, well, macroevolution is just change at or above the species level. Well, no, because when you actually look at how new species come about at a genetic level, it's due to changes in or shifts in heterozygosity, which is more allelic variability, more levels of genetic diversity, to homozygosity, less allelic variability, less genetic diversity. That's what we see. Those are downhill changes, although we are getting changes, new varieties. It's downhill, it's not uphill. These are horizontal changes. These are not vertical changes. And so in the picture here, scenario 1, DNA copies different, a heterozygous state, right? Scenario 2, DNA copies are the same, homozygous state. God said to be fruitful and multiply. Did he really want that to be carried out through cloning? Because that's what you would get with created homozygosity. No, it makes sense that God would have created Adam and Eve. This would apply universally to the created kinds with pre-existing diversity, with created heterozygosity, in order that we could truly be fruitful and multiply. So this makes sense theologically, and it also makes sense scientifically. Now, I've done a series of videos addressing Gutsa Gibbon on these issues. I recently teamed up with Apologetics 101, and I responded to her in detail, word for word, from a video she did on junk DNA. And so we've got the critics on the defense. The ball is in their court, and they've yet to address us thoroughly and comprehensively. I've also responded on the chromosome 2 issue. Okay, so with that, we're going to get right into dismantling junk DNA, as we have now set the foundation for what will be discussed. Okay, so right into the junk DNA issue. There is a key difference when it comes to the ancestry models. And so the evolutionist expects the genomes of living organisms to reflect these long evolutionary histories and processes, descent with modification. So they expect a lot of evolutionary baggage, evolutionary leftovers, genetic mistakes, genomic fossils, viral fossils. Is that what we find? Because the creationist expects treasure rather than junk. And so that is why DNA function is an excellent way for us to answer this question of ancestry in a way that helps us discriminate between the models. Because if the genome is mostly junk and only about 5 to 10% functional, then that would be a point for the evolution side. But if the majority of the genome is actually functional, say above 65%, then that is a major point for the creationists, because only the creation model can explain that, especially in light of the mutation accumulation problem. Deleterious mutations are pouring into the genomes of living organisms rapidly. Most mutations are unselectable, meaning selection can't see them. They're nearly neutral, meaning they're neutral to selection. Selection can't do anything about them, but they're nearly neutral because they have very small effects on organismal genetic content. Small effects on fitness, like a single spelling mistake in a book the size of an encyclopedia, or a single rust molecule on a car. On their own, they're inconsequential. But as they accumulate over time, they degenerate, they damage, they destroy. Mutations are the destroyer and not the creator. Mutations are destruction, not construction. And so, over time, these mutations accumulate and they destroy. They put shelf lives on genomes. And that's why the evolutionary community has had to largely invoke junk DNA, because they need to argue that most of the genome is just non-coding, neutral junk. And so when these mutations, the mutation rate's about 100 new mutations per person per generation. That's a lot. Most of those being deleterious. That is detrimental to the health of populations. So the evolutionist wants the majority of the genome to be junk in order that most of those deleterious mutations are not deleterious, because they hit junk regions. The junk regions absorb those deleterious effects. There's no harm. But that's not the case. The more functional the genome is, the more problematic it is for the evolutionist. Just real quick, non-coding DNA. Basically, your non-coding DNA functions in regulating the coding portions of the genome. And so here's just a few. We're going to go over a lot of these in detail. Regulating expression of genes, where you got DNA to RNA to protein. DNA transcribed into RNA, translated into protein. The process of turning a gene on to produce an RNA product or a protein product. Gene expression, the process of gene expression. You've got gene regulation. So regulating this process that turns genes on, turns genes off. Maintaining genomic integrity, providing structural elements to chromosomes, switching on and off genes, replication, so on and so forth. So there's a lot of known functions for the non-coding areas of our genome. Because we understand most of our genome based on the ENCODE project and future projects. Most of the genome has tremendous evidence for what's called biochemical function or activity. It was in 2012 here that the ENCODE project came out with these remarkable findings that indicated upwards of 80%, as you can see here on the slide, of the human genome was active to some extent. This is detectable biochemical function. This is the definition they used. And it was very surprising. It was flabbergasting to discover that the genome consists of only about 20,000 genes. Okay, this is an estimate, but it was not an anticipated number. What we now know and what we now understand is that a vast portion of the human genome consists of what? Non-coding DNA sequences, non-coding DNA elements, as in sequences that don't directly code for protein products. Because again, you have DNA transcribed into RNA, translated into a protein, but you don't always get that translation aspect of this process. Okay, protein synthesis. No, you most frequently get DNA transcribed into RNA, an RNA product, and that product is functional. It's useful. It's not junk. And so you can see here, these data enabled us to assign biochemical functions for 80% of the genome. Now, the best definition of function, I would argue, biochemical interactions that have consequences for the cell. When we study the interactions between the systems of a cell, because your evolutionary community, they try to attack the biochemical function definition that the ENCODE project used. Well, here's your best definition: biochemical interactions that have consequences for the cell. And that's what we find in the genome, because as we study interactions between the systems of a cell, remember, the genome is not just this linear string of letters, ATCG, adenine, thymine, guanine, cytosine. Okay, it's not just this linear string of letters. No, the genome is multidimensional. It's a 4-dimensional code with chock full of meta information, multiple overlapping codes. It's nested, extremely functional. But a lot of evolutionists, they have a 1-dimensional, unsophisticated, unimpressive view of the genome. And so as we study these interactions between the systems of the cell, we find a lot of activity. This is important activity. And so if there are interactions taking place, then we can confidently conclude there's a function. We have to look to transcription, but not only transcription, transcription factor binding, protein binding to DNA, histone binding, histone modifications, three-dimensional interactions, because the fourth dimension's time. So we have a four-dimensional genetic code. And so we look to three-dimensional interactions within the code. We look to RNA binding, and these are all what? Biochemical assays. And so if there are interactions that are taking place in the cell, and these interactions have outcomes or consequences, then we can conclude function, because we should expect treasure. The evolutionary mindset hinders the advancement of science. And so the biochemical definition is a great definition. It's a great way for us to approach the genome in terms of looking for function. So, desperate evolutionists, they don't like this evidence, they don't like this data, and so they've argued that most of the transcription taking place in the genome is merely spurious, or it's nothing more than genetic noise. It's not useful activity. But this appeal doesn't make much sense. It's anti-science, it's pseudoscience. Since we know, we've recognized, studying these issues, that transcribing a portion of the genome is a very energy-intensive process. It requires ATP. And every time a nucleotide is being added to a growing RNA chain, you are consuming what? A great deal of energy. And so you have all these transcripts in the genome being produced. ATP is being used. Energy is being utilized. But yet it's all spurious. It's just noise. No, that doesn't make any sense. That's not intuitive. That's not science. It's weak. It's a weak rescue device. And the evolutionists still employ it even though it's been demolished. These transcripts, if this was just noise, these transcripts would clutter up the interior of the cell if they weren't actually playing any real operational role. And so the question is 80% of the human genome biochemically active? And does this comprise real functional significance? When it comes to the transcription, they've argued that it's now about 60%. Okay. Activity-wise, there's about 80%. Way too much for evolution to be true. Evolutionists need the function to be around 10%. Even that, I would argue, is still too much in light of deleterious mutation accumulation. So, point I just touched on, why it is real activity, not just spurious. Transcription, an energy-intensive process. It's a resource-intensive process. These nucleotides, as I've pointed out, they're being utilized to produce RNA molecules that end up, according to the evolutionists, being noise or spurious. That's very problematic. Since this is an energy-intensive process, since it is a resource-intensive process, and the genome is using up a large amount of energy and resource to produce these transcripts, they must be doing something, because that would be wasteful. It would be a wasteful process, and there should be mechanisms to minimize that process. This type of, if it were true, this type of unnecessary transcription would most certainly be dealt with through various mechanisms. If this were the case, this would be dealt with. You're trying to tell me, evolutionists, that evolutionary processes can take a single-celled like ancestor into a multi-celled ancestor, that multi-celled ancestor or organism into a fish, amphibian, reptile, mammal, and then a higher mammal like a human. Evolutionary processes could take a fish to a fisherman. Evolutionary processes can take goo to you, pond scum to people, but they can't remove all this junk in order to declutter the system. Decluttering is good. Decluttering your house, decluttering your car, decluttering your drawers and your closets with your clothes and your just socks and all this stuff building up the backseat of your car, especially when you're in school. I remember being in college, it was a breath of fresh air to declutter the car because it reduces productivity. It's stressful, causes anxiety, just like in the genome. And so these processes, if this really were the case, should be removing all of this junk and baggage, because it would be wasteful of energy and resources for the cell. Next point. Transcription factor binding. So a lot of this is technical. It's heavy on genetics. And I like to define the terms we're discussing here. Transcription factor binding. Okay, well, we have these transcription factors. They're proteins. And what they do essentially is regulate the transcription of genes. Remember we talked about earlier, DNA transcribed into what? RNA, and then translated into a protein. So you have these proteins, they're regulating the transcription of genes, as in they're copying into RNA on the way to making a protein through translation, through the ribosome, okay? And these transcription factors, well-designed DNA elements, they actually help to ensure that the right genes are expressed in the right cells of the body at the right time. It is amazing what is going on in the genomes of living organisms. And RNA polymerase can attach to the promoter. A promoter is a genetic switch within the genome that initiates transcription. Think of your light. You come downstairs, the light's off, you turn it on, you got light in your basement. So these are genetic switches in the genome. And the RNA polymerase can actually attach to one of these switches, one of these promoters, but only with the help of, guess what? These proteins that we call transcription factors. And these binding sites, okay, these proteins that can bind to sites within the genome to help with transcription, they are often close to a gene's promoter. And so everything's working together. Everything is working as if it was designed to work that way, rather than through blind evolutionary processes. Now, here's the issue for the evolutionists. If you have high levels of non-specific binding, let's say this were all noise or mostly noise, what you are doing is you are titrating out transcription factors, these proteins. This means you won't be able to get the transcription factor binding that is actually necessary in the genome. Random binding, non-functional binding would what? It would gum up the operations within the cell. Most of this binding that has been discovered through these various projects, like the ENCODE project, must be serving a purpose, or else operations within the cell would be extremely messy. This would be problematic. This would be an issue. Again, this is an energy-intensive process. If operations in the cell were mostly just noise, as the evolutionists are arguing, this would be a waste of energy. It would be a waste of resources for the cell. And so we have transcription, an energy-intensive process, and transcription factor binding. Two major reasons why this function has to be real function, not just noise. If transcription factors are just randomly binding all over the genome, that would simply muck up the systems of the cell. This would actually create what's called an interference problem. The interference problem is what would gum up operations in the cell, because there's only so many copies of a transcription factor. These proteins are not just unlimited of them. Okay, and so there's only so many copies of these transcription factors that are going to be produced. And if these transcription factors were merely binding to random sites in the genome, random locations in the genome, then there wouldn't be enough transcription factors available, as I pointed out, to actually do the binding that is necessary for these transcription factors to properly function. And many of these points are important to reiterate in order for the evolutionists to understand, to get it in their brain that no, you're not related to a strawberry. No, most of the genome is not junk and non-functional. These arguments are weak. And that's why they typically dodge or avoid answering these challenges or our rebuttals to their, what I would argue, are rescue devices. As we can see, there are numerous good biochemical reasons to believe or to understand that transcription factor binding is not just noise. It reflects functionality, real functionality. Here's a few more key points. Redundancy. Redundancy is good design. When God created living creatures with front-loaded genetic diversity, as we talked about earlier, he front-loaded the potential for incredible amounts of adaptability into their genomes. And evidence of design diversity is function, as we've pointed out, but it's also redundancy. Engineers, they know how important redundancy is. Redundancy is basically having backup. If a gene or a DNA element is mutated or damaged, the backup genes will help significantly. Let's say you have a group of genes that are important in a certain function. Well, if you only had one of those genes and it's destroyed through mutations, then that's it. But if you have a multitude of those genes, when one's hit or a second one's hit or even a third one is hit, you have backup. You have redundancy that can still do the job. Okay? And that is what we find in the genome. And we find this in human design, human engineering. Think of your tire, spare tire. You have 4 tires, but you also have a spare tire. The evolutionists would look to that as, oh, that's evolutionary leftovers, that's junk. No, if you get a flat tire, you're in an environment where you get a flat tire, then that spare tire is going to come in handy. You're going to be glad you had that spare tire. That is thinking ahead. That is forward thinking. And forward thinking mechanisms points back to a forward thinker. We know that that spare tire was put there in the vehicle that you purchased because of the foreknowledge of the engineer that built the car. He knew, hey, we're going to put a spare tire because the driver might get in a situation where they need a spare tire. And thank God that there's one there, because redundancy is good. There's another concept or point called over-design or over-engineering. And this is good. When you design, for example, or build a bridge, let's say, you don't just build a bridge to hold up the weight of the exact number of cars that you expect to drive on the bridge. No, you're going to build it 10 or 20 or 30 times stronger than the number of vehicles you actually expect to be driving on the bridge. If you think there's going to be 1,000 cars driving on the bridge, you don't design and build the bridge to hold up exactly 1,000 cars, and then boom, you get 1,001 and the bridge collapses. No, you build it 30 times stronger than what it can hold. And that is good design. That's called over-engineering or over-designing. And this kind of design, what it adds is robustness, it adds reliability, and this is the way genomes are designed. And many organisms, like especially in the plant world, they have more DNA than humans do, because they need a lot more redundancy because of the environments they live in. And so that's why they have a lot more DNA oftentimes, is because they're over-designed to withstand the harsh environmental conditions that they exist in, or they might need more genetic elements for adaptation. And so the amount of DNA doesn't necessarily reflect how complex the organism is. And it doesn't mean because, well, this specific organism, an onion, or this specific salamander or fish has 20 times more DNA than humans, means it's non-functional, it's junk. No, it means it's over-designed for increased robustness and reliability, or the ability to adapt. Okay. When we step outside on a cold day as humans, we have coats we can put on. We can wear heavy shoes, heavy boots. But a lot of organisms in the wild, including plants, they can't exactly do that in these harsh conditions. And so they have a lot of front-loaded redundancy. They have a lot of over-design or over-engineering to help them survive. It's survivability, but it's all there for a reason. It is all important in terms of function. We expect treasure. There's another thing called epigenetics, which has to do with adaptability. And so, because it has to do with adaptability, we know that as biological organisms, as they move or as they navigate through different environments, genes, pre-existing genes within the organism's DNA can be turned on or they can be turned off depending on what is necessary to survive within the specific environment. Genomes, organisms have millions of DNA switches that are just waiting to be turned on or off through the right environmental stressor or stimuli. And so epigenetics, above and beyond the DNA, this is amazing sophistication at play here, amazing evidence for design. Epigenetics results in a change within the activity of the genes. And because of that change, you can get rapid adaptation, rapid change. But it wasn't through long evolutionary processes. It was because that ability to adapt was front-loaded. It's pre-existing. And we're seeing this more and more, paper after paper, suggesting this kind of rapid, quote-unquote, evolution. But it's actually rapid adaptation. And it's an amazing time to be a biblical creationist because more and more the genetic data mounts that demolishes common descent and confirms design diversity. Then we've got embryological development. So many genes, they're only functional during various stages of the developmental process. Okay, going from zygote to fully formed human being, the baby in the womb. ERVs, for example, many ERVs are only functional during different parts of embryological development, where once they're done doing their job, they're turned off. Many pseudogenes, for example, many non-coding RNA genes are only functional under certain circumstances. And so you could knock those out, but if they're not turned on, there's not going to be an effect. And so you need a almost unlimited sensitivity in what's called knockout tests to determine what is truly functional and what isn't. And so the extra complicated, the amazing process of embryological development, a lot of these genes that the evolutionists say are junk, they're actually involved in embryological development. But after birth, those genes are either repurposed for other functions or turned off to maybe be called upon again in case of the right environmental stressor or stimuli. It doesn't mean they're not functional. Okay, just like you have the construction of an amazing building, let's say, or a house. During the construction of it, you're using all sorts of tools, you're using all sorts of mechanisms and things that you're not going to be really using. They're going to be dormant once the building is finished. And so you have all these construction workers working full-time, getting the building or the house designed, up and running. And once it's up and running, all of those tools, great multitude of sophisticated tools, they're no longer being used. Well, it doesn't mean they're junk. They've already been used and utilized for the design, for the building of that specific building, okay? Or another good comparison or illustration, let's say you're watching a play. You have a play. We have all the actors and actresses up on the stage. But is that all that's taking place for this play to make this play possible? No. You have behind the scenes, people working the lights, the directors, the writers, the producers, sound effects designers, okay, music, things like that, especially in a movie, right? When you end a movie, you have 5 minutes oftentimes of just all the names of people that worked on the movie, that made the movie possible, behind the scenes workers. And so a lot of what we see in the genomes of living organisms, especially in the non-coding regions of the genome, are behind the scenes workers. What's going on behind the scenes. And that's why your protein coding genes oftentimes turn out to be more similar across creatures. Okay. But then your non-coding regions, providing the metadata for the protein coding genes to function, those tend to be more taxonomically restricted, less similar across creatures. Okay. So yes, embryological development is something that the evolutionary community underappreciates. What's really going on, how sophisticated this developmental process is. So as we could see here, I mean, I could go through, as we start to wind down the presentation, paper after paper, going over the amazing functionality of the genome, junk DNA overturned, scientists discover a role for junk DNA, redundancy of the genetic code enables translational pausing. So even your redundant elements, right? There's redundancy in coding for a protein. Well, it turns out this wobble position 3, it isn't just there to tolerate error, third position codon variation, it helps slow and speed processes in the cell. So even there, we've got function, which means when they get hit with mutations, yes, it will be nearly neutral. There will be some effects. Just like the treading on your tires. As you're driving day by day, it's almost unnoticeable as the treading wears down. Doesn't mean it's completely neutral, because as time goes on, if you actually look at day 1, your new tires, and then day 150 or day 100, depending on how much you drive, the treading is going to wear down to the point where you need new tires. And so there's a lot of redundant elements in the genome where at first, if they're hit with a mutation, there's backup code. There's a lot of redundancy to help the genome tolerate error. But over time, as time goes on, the more those redundant elements are destroyed and worn down, the more harmful the effects are. So we've also got pseudogenes. Pseudogenes, we now know, are functional DNA elements. They are necessary to sustain healthy life processes in the cell. You've got theistic evolutionists like William Lane Craig that say some of the best evidence for why he accepts evolution. You don't accept evolution, you believe, capital B, in evolution, is because of pseudogenes, so-called genetic mistakes. They'll argue, well, we've got these genetic mistakes between humans and chimpanzees, and so we must have inherited those mistakes from a common ancestor, rather than those mistakes coming about independently in both lineages. So the question is, why do pseudogenes look like they're healthy protein coding counterparts? Well, we now understand, there's a few papers here, that the pseudogene expression actually helps to regulate the translation of its protein coding counterpart. They work together. Notice from this paper in 2010, together these findings attribute a novel biological role to expressed pseudogenes, as they can regulate coding gene expression and reveal a non-coding function for mRNAs. This is amazing. Many of the roles these pseudogenes have, just like ERVs, are dependent upon their similarities to their protein coding counterparts. Just like ERVs, many of their functions, like functioning in the immune system, fighting off exogenous retroviral infections, requires them to look like viruses, to have those structures, the GAG, the POL, the ENV, the LTRs. They require that to carry out their roles. Just like the pseudogenes, the non-coding counterparts have to look like their protein coding counterparts to carry out their roles. Okay, so basically you have these long non-coding RNAs. They're not coding for a protein. They enter various transcription networks. And all of this works in combination because we have an incredibly elegant system within the genomes of living organisms. You have different kinds of pseudogenes, processed pseudogenes, unitary pseudogenes. Unitary pseudogenes, they're actually playing a functional role in gene regulation since they are part of functional transcription families. You have pseudogenes that don't have a promoter, or the promoter's damaged, the evolutionists will say. No, what we see in terms of the differences, the properties of these pseudogenes, they're designed that way. Evolutionists are assuming that this is due to mutations because they assume all differences are due to mutations over time. No, we find pseudogenes that aren't transcribed actually function in chromosomal stability or architecture, where if you remove them, then it messes up recombination, it messes up chromosomal integrity, messes up chromatin. So they're there for a reason, just because they aren't transcribed. So transcriptionally silent pseudogenes and even transcriptionally silent ERVs also have a functional role. It's function, function, function. And this is what we predict. We don't find junk DNA. We find treasure. We find function and a lot of it. There's also something called the decoy hypothesis, the pseudogene decoy hypothesis. And basically the RNA that's produced by the pseudogene. Remember, the evolutionists assume that this gene once was able to code for a functional protein, so translation was possible, but now it doesn't. Many of them produce an RNA product, but it's because they're broken. No, they're designed to do what they're supposed to do. And so the RNA that's produced by various numbers of these pseudogenes can act as a decoy for things that might actually target the correct RNA from the protein coding counterpart. Okay. And so they might target the correct RNA for degradation from the protein coding counterpart that would what? Well, it would, as a result, increase the lifespan of that specific mRNA and therefore increase the amount of what? Translation. Because the protein coding counterpart can translate DNA transcribed into RNA. The protein coding, the non-coding counterpart, produces the RNA. The protein coding counterpart can translate it into a protein. And so this decoy mechanism helps preserve the lifespan of that specific mRNA and increase the amount or effectiveness of translation. And so these are amazing functions right here that actually are dependent upon the pseudogenes sequence similarity to the protein coding versions. And this is just a snapshot of the overall functionality of pseudogenes, because I am working on an all-out presentation. I want to put together the most comprehensive presentation on pseudogenes available today, because the evidence really is that amazing. So I did a series a couple of years ago with Dr. Fuzz Rana addressing these issues, and we've discovered so much more even since then. And we discussed the many functions of junk DNA. A lot of the so-called areas known as junk DNA act as a mutational buffer, maintain 3D structure, regulate histone binding. There's something called the nucleoskeletal hypothesis. So basically what that is you have DNA, and it's helping to regulate the volume of the nucleus, the nucleoskeletal hypothesis. And notice here, DNA content determines nuclear volume. The ratio of nuclear volume to cellular volume is a tightly constrained parameter. Pseudogenes, as we pointed out, gene regulation, decoy mechanism, some even coding for functional proteins. Lines, long interspersed nuclear elements is what that stands for. Then you've got signs, okay, and X chromosome inactivation, monoallelic gene expression, signs function in stress response, gene regulation. Introns, same thing with introns. They allow for alternative splicing. Stress response. A lot of these DNA elements act in terms of cell stress responses. They function in the immune system. ERVs, they have antiviral functions. They function in tumor suppression, these ERVs. There's a superhero protein. It's called the P53 protein, known as the guardian of the genome. And these work with the ERVs. A lot of these ERVs, they can give the cell the appearance of being invaded. So a transcribed ERV can give the cell the appearance of being invaded by a virus. And so the immune system targets that cell, that tumor cell, with more destruction. And as a result, it works in combination with the P53. It destroys tumor cells through viral mimicry. And so the ERV has to look like a virus in order to carry out that function, or else that function wouldn't work. Here's a paper that talks about multiple knockout mouse models reveal that these RNAs are required for life and brain development because they knock them out. And a lot of the knockout tests revealed that they were lethal. They killed the mouse, or the mice were diseased. But again, many non-coding genes are only functional under certain circumstances. There's also a lot of over-design, a lot of over engineering, a lot of redundancy. So just because you knock out a gene in a mouse and the mouse still lives, or even if the mouse isn't diseased, doesn't mean that that gene is not functional. This is just a fact. This is reality. And so now we're getting into paper after paper on everything to do with the world of junk DNA, endogenous retroviruses. And you'll find, notice this, long disregarded as junk DNA. So they admit that a lot of these DNA elements were once thought of as being junk DNA or genomic dark matter. Endogenous retroviruses have turned out to what? Represent important components of the antiviral immune response. They not only regulate cell immune activation, but may even directly target invading viral pathogens. So they're like these antiviral programs in our genome. And so I've done entire presentations on ERVs. I wanted to focus on the bigger picture here, plus give a little bit of data on pseudogenes, ERVs. But again, without these units, we would not exist. Notice this one. HERVs, the H stands for human, so human-specific ERVs, play important roles in physiology, fetal development, and human evolution. If the accidental infection of a mammalian ancestor by an exogenous occurring from outside, rather than occurring from within, the endogenous retrovirus, had never occurred, the placenta and the mammals that produce it, including humans, would have never existed. I like to exist. Hope the evolutionist likes to exist. Hope the listeners like to exist. And so if we didn't have these DNA elements, we wouldn't exist. And so if we share these elements with the biological world, it's not because of common descent, it's because of common design. Notice this, paper after paper. I could spend 3 more hours going through just the dozens and dozens of papers that overturn the junk DNA paradigm. ERVs, pseudogenes, transposable elements, ALU sequences. HERVs appear to play important roles, as we pointed out. Physiology, fetal development. In the mouse, there's a certain class of retrotransposon. The mouse is developing, you snip it out. The mouse is developing. Once you snip it, it dies. Because development depends on these retrotransposons. Here's what I was talking about earlier. This paper, 2021. ERVs are emerging as promising therapeutic targets in cancer. This specific HERV, HERV-H, can modify the higher order 3D structure of chromatin during early primate development. They're necessary for life. Endogenous retroviruses as a what? Unexpected. The evolutionary community, they could say, oh, this is expected. No, they didn't predict it. I want to see where they predicted it. Because in paper after paper, they say this was unexpected. They were surprised. Endogenous retroviruses as an unexpected ally in the regulation of human immune systems. LTR functionality. Notice this. I was saying this earlier. ERVs have become integral parts of immune defense mechanisms and help to fight off invading viral pathogens. And guess what? They require that sequence similarity to exogenous retroviruses to carry out their many functional roles. And that wraps up my presentation, guys. I know there's a lot there, but I really wanted to set the foundation for the ultimate dismantling of junk DNA. This actually wraps up our Defending Genesis 2023 conference. It's been a blast. We've had 11 speakers and 10 shows. And so please, if you have enjoyed this conference, share it around. We have had an epic week of shows. We have had, here we go. We started the week with Dr. Dan Biddle, Noah's Flood. Matt gave an excellent presentation on Noah's animals on Noah's Ark. John Mackay and Joe Hubbard, of course, were here. Great presentation here. Great presentations all around. Steph Harima, Helmut Welk, presentation on Adam and Eve. Professor David McQueen, Redefine Living. We had an epic show with Dr. Dino. This included an open mic. And my show here, Junk DNA Debunked. So again, 10 shows in 1 week, over 15 hours of content. I think about 17 hours of content. So we want to do this stuff public. We know the body of Christ needs this information, and we're willing to put in the work. We're willing to engage the critics. And so we're doing this full time right now. If it is on your heart to support the work that goes into setting up these kinds of conferences, that goes into hosting these conferences, the research itself is a full-time job, then feel free to donate, support what we're doing here. And you can find those links in the description box. It helps greatly with our continued efforts. It helps us to, especially myself, to continue doing this full time, to put on some epic interviews, debates, and conferences like this for everybody, especially as we move into 2024. I'd love to do some in-person stuff. And the more that we implement, the more that we integrate into the ministry and what we're doing, the more costly it becomes, of course. And so we are a listener-supported ministry, and we're willing to put in the work, and we're willing to provide you the most up-to-date material and the best content available. And so again, if it's on your heart to support us, please do consider it, as we would greatly appreciate that. And with that, we're wrapping things up. God bless all. Thank you so much for tuning in. I hope you enjoyed this. The critics, my friends, they're on the defense, and the ball is in their court, because it's not such a good time to be an evolutionist, but it is a great time to be a biblical creationist. God bless.